Hi everyone, I'm Janani Sampath. I'm a postdoc at the Pacific Northwest National Lab and the University of Washington, and I'm excited to virtually talk to you about my work on sequence control polymers for biomineralization. A very interesting phenomenon that occurs in nature is that of biomineralization. It's the process by which living organisms produce multidimensional mineral structures and is something that occurs in our bodies with the production of bones and teeth. It's also observed across the animal kingdom. The picture that you see over here are these exquisite hierarchical silica nanostructures made by diatoms under ambient conditions and neutral pH using an orthosilicate precursor. In contrast, state-of-the-art techniques to manufacture well-controlled nanostructures in the lab involve high-temperature solvents, and this process is often energy-intensive and harmful for the environment. And so people are constantly thinking of ways to make this process more efficient while maintaining good control over the mineral morphology. It was discovered that in diatoms, the silafin protein with 7 peptide segments was responsible in mediating the formation of silica nanostructures. Within silafin, the 19 amino acid segment R5 was found to be the most crucial part, as it has a similar efficiency as the parent protein, where they both produce nice spherical silica nanospheres in vitro. Studies also found that natural R5 extracted from the cell wall of diatoms actually contain numerous post-translational modifications at the site of serine and lysine side chains. Post-translational modifications are biochemical modifications that occur at the site of an amino acid after a protein has been synthesized in the cell. Investigation into the role of these post-translational modifications revealed that they are crucial to the precipitation of silica. This graph shows the amount of silica precipitated as a function of peptide concentration. And so the red curve corresponds to natural R5 with a whole lot of post-translational modifications. And this system shows a high silica precipitation activity. On the other end, the green curve with no silica precipitation activity corresponds to synthetic or wild-type R5 with the same amino acid sequence, but stripped of any post-translational modifications. To get the wild-type R5 to precipitate silica, the presence of phosphate buffer is required. And we see that there is a concentration dependence to this as well. And so R5 needs post-translational modifications or the presence of phosphate buffer to precipitate silica in vitro. Regarding the mechanism of peptide-mediated mineralization, there is some consensus that prior to silica precipitation, these peptides form aggregates, which serve as sites for condensation of the silica precursor. So if the peptides don't aggregate, then it means that they can't precipitate silica. And it's posited that both phosphate ions from the buffer and the phosphorylated side chains serve the same purpose in bridging the positive groups between the chain. No one has yet been able to capture the structure of these aggregates. And so to test the hypothesis that aggregation is a prerequisite to mineralization, we decided to investigate 7 systems, 5 of which have been studied using experiments, and 2 are controls for our simulation. Without getting into the particulars, we chose these systems as they spanned a large design space and would allow us to draw strong correlations between post-translational modifications, solution chemistry, and peptide aggregation if it exists. We characterize the ability of 2 peptides to form dimers as an indicator of aggregation. If they can't dimerize, it's unlikely that they would be able to form higher order structures. In this study, we used all atom molecular dynamics simulations with explicit water molecules and ions to capture relevant interactions between peptides, ions, and water, including electrostatics, hydrophobic interactions, and hydrogen bonds. The aggregation or dimerization behavior that we are interested in capturing occurs over very long times. And to overcome this limitation, we enhance the sampling of our system using metadynamics. In metadynamics, systems are often represented by a few low-dimensional descriptors as a function of atomic positions known as collective variables. So in the case of two molecules coming together to form dimers, a good collective variable would be to track their centers of mass. When thinking about a one-dimensional free energy profile with two states A and B separated by a large energy barrier, systems are often trapped in one or the other metastable state. And due to this barrier, crossing from one state to another will take a prohibitively long amount of time. In metadynamics, an external bias potential in the form of a Gaussian is deposited periodically, forcing the system out of metastable states, allowing it to sample many configurations. And in this way, we're able to get good sampling and thoroughly explore the phase space. So the first set of results that I'm going to show you today is the dimerization of a wild-type R5 peptide with and without phosphate buffer. The graph is a free energy profile. On the y-axis, we have the free energy of dimerization, and on the x-axis, we have the center of mass distance between the two peptides. The red curve shows that the peptides have a strong propensity to bind when phosphate ions are present. And this can be seen by this deep minima at small peptide-peptide distances. When there are no phosphate ions in the system, we see that the peptides do not dimerize, as shown by the blue curve, with no minima at short interpeptide distances. This agrees well with experimental results, where the red system mineralizes silica, but the blue system does not. Cluster analysis reveals that electrostatic interactions dominate the peptide dimerization. However, once they come together, the dimers are stabilized by hydrophobic side chain interactions. Next, we explored the effect that the location of phosphorylation has on dimerization and whether or not it matters which side chain is phosphorylated. We find that the dimerization landscape changes with the location of serine phosphorylation. When the serine near the C terminus of the peptide is phosphorylated, the free energy of dimerization is higher compared to when the serine near the N terminus is phosphorylated. This variance is because of the different hydrophobic groups that interact with each other once the dimer is formed, as can be seen in the cluster analysis. Both these mutants show silica mineralization in experiments. However, the morphology of the minerals at the end is different. And so these, for examples, illustrate the strong correlation that exists between a peptide's ability to dimerize, which we've quantified in our simulations, and its mineralizing capability as seen through experiments. And so the overall goal of studying biomineralization is the ability to control it and to understand how a certain molecular sequence can lead to a specific mineral morphology. In this regard, peptides or N-substituted lysines are ideal candidates to understand structure-function relationships because of the diversity of side chain chemistries accessible through peptides. As the side chain in a peptide is attached to the backbone nitrogen atom, instead of the alpha carbon atom as in the case of peptides, assembly through backbone hydrogen bonds is eliminated, leading to interactions only through the side chains, which in turn can be rigorously tuned and controlled. And so we hypothesized that since R5 is intrinsically disordered with no secondary structure, its ability to form aggregates is dominated by side chain groups and not due to backbone interactions. And if we were to design a peptoid analogous to the R5 peptide, it should mineralize silica as well, as long as it's able to show a similar aggregate behavior. And so to test this, we first modeled the R5 peptide and calculated the free energy of dimerization with and without phosphate ions. From the free energy profile here, we see that similar to the peptide, the R5 peptide doesn't aggregate without the phosphate ions either. Closer inspection of the single peptide conformation reveals that hydrophobic interactions dominate, causing charged positive side chains to be exposed in solution. Without the phosphate ions, the peptides tend to repel each other due to like charges. Upon the addition of phosphate groups, dimerization occurs. And so we took these results to our experimental collaborators who at the same time were trying to design a silica mineralizing peptide. Our hypothesis was that the peptide should mineralize silica as its dimer structure is very similar to the R5 peptide, and the backbone hydrogen bonds do not seem to have an effect in its mineralizing ability. And so the lack of it in a peptide shouldn't be an issue. Upon synthesizing this new peptide and carrying out similar analyses on the peptide and the peptoid, we see remarkable similarities in the mineralizing function across the two systems with homogeneous silica nanospheres, proving that the ability to dimerize is a strong indicator of mineralization for peptidomimetic materials. And so in conclusion, we found that aggregation is an indicator of biomineralization in R5 by extensively testing many systems using molecular dynamic simulations. We've been able to learn design rules from nature and apply this successfully to a synthetic system. In the future, we want to test the following mutations to see if we can draw connections between molecule sequence and mineral morphology, and eventually take advantage of the large design space offered by peptides to regulate mineral formation. And so with that, I'd like to thank my advisors, Jim Fentner and Chris Mundy, as well as my experimental collaborators, Chunlong Chen and Francois Banex. I'd also like to thank the Department of Energy for funding this study through the EFRC that I'm a part of called CSAS. And I'd be happy to take any questions if you have any. Thank you.